In this class, we will discuss about endoplasmic reticulum and Golgi bodies. These two are cell organelles. As they are having so much similarities, we are discussing both together. This is the simulated view of a cell. This is the nucleus, yellow color. Close to this, you can see endoplasmic reticulum. Then you can see the Golgi bodies, and this is the cell membrane. And if you see the scanning electron micrograph, this is the nucleus, and you can see the endoplasmic reticulum proximal to nucleus. Now, what is endoplasmic reticulum? It is a membranous network. They are formed of membrane bound structures, and all these structures are having internal cavities. Endoplasmic reticulum is formed of three kinds of structures: cisternae, vesicles, and tubules. And it is first introduced, or the term endoplasmic reticulum was first introduced by Porter, and they are distributed throughout the cytoplasm outer to nucleus. Coming to the function, they do protein and lipid biosynthesis, and they act as the cytoskeleton of the cell and cytoplasmic vesicular system. A tubular system, vesicular system means it's a tubular system which can carry the materials within the cell for a lateral transport. And this vesicular or vacuolar system extends from cell membrane to nuclear membrane. So, from the cell membrane, this endoplasmic reticulum is having a connectivity till nuclear membrane. So, within a cell, it acts as a conducting channel from the nucleus to cell membrane. And if you see the structure of a diagrammatic sketch of a cell, you can see these are the endoplasmic reticulum, and this is the central nucleus, and it is somehow connected and it will reach till the cell membrane outside. The membrane of endoplasmic reticulum is trilaminar, and they have connectivity to plasma membrane, Golgi membrane, and nuclear membrane. The endoplasmic reticulum always acts as a passage for the transport of secretory products. Depending on the presence or absence of ribosome on the surface of endoplasmic reticulum are classified into rough endoplasmic reticulum or granular endoplasmic reticulum, and smooth the other endoplasmic reticulum without ribosome on the surface are called smooth endoplasmic reticulum. You can see the image. This is the rough endoplasmic reticulum, where the surface of the cisternae are having lot of ribosomes studded on it, and this is the smooth endoplasmic reticulum. The difference is rough endoplasmic reticulum have ribosome on the surface, and the smooth endoplasmic reticulum doesn't have the ribosome on the surface. And chemically, the endoplasmic reticulum is made up of lipids and protein, or the membrane itself is made up of lipids and proteins, and hence it is called a lipoproteinaceous membrane. The presence of the enzymes like NADH cytochrome C reductase, NADH diphosphorase, glucose 6-phosphatase, and Mg activated ATPase are seen within the endoplasmic reticulum. And coming to the different structural components of endoplasmic reticulum, as I told in the very beginning, endoplasmic reticulum is formed of three kinds of structures. One is called cisternae, second one is called vesicles, and third one is called tubules. First one is cisternae, and you can see the image out here. They are like flattened sac-like structures. So cisternae are elongated, flattened, unbranched sac-like structures, and they are often arranged in a group as a bundle. And just we'll go back to the previous image. And this is how cisternae is observed. They are grouped as a bundle. They are seen together as a bundle. Cisternae are observed as bundles like this. one above the other on the surface, and they are having a diameter between 40 and 50 microns, and these cisternae are rough due to the presence of ribosome in the case of rough endoplasmic reticulum, but otherwise it is smooth in the case of smooth endoplasmic reticulum. And the second components are called vesicles. Vesicles are rounded or oval structures with a diameter of 25 to 500 microns. They are the second component of the endoplasmic reticulum. Normally, vesicles are seen at the end of the cisternae and tubules. So these are the cisternae and these are the tubules. Often these vesicles are seen at the tip. It's something like releasing from the, it is observed like releasing from the endoplasmic reticulum as free bodies. They are called vesicles. And even vesicles are observed free in the cytoplasm, which says us that these vesicles are pinched off from the endoplasmic reticulum. And third structure is called tubules. Tubules are called branched tubular structures. Their surface is smooth walled, so they are not having ribosome on the surface. Their diameter is between 15 to 100 micrometers, and often these tubules are present in the striated muscle cells. They are observed commonly in, or always they are seen in the striated muscle cells. And coming to the functions of endoplasmic reticulum, they give mechanical support to cell, and hence they are called a cytoskeleton. As they are connecting the cell membrane to plasma membrane, they act as a lateral circulatory system. Normally, protein synthesis is carried by ribosome, and endoplasmic reticulum act as the site of attachment of ribosomes. The synthesized proteins from ribosomes are stored and transported by endoplasmic reticulum. They synthesize cholesterol and steroid hormones. And the endoplasmic reticulum present in liver, they do detoxification, and they are capable of doing lipid synthesis and also glyconeogenesis. Glyconeogenesis is a process by which glucose is produced from lipids. And this is the structure and function of endoplasmic reticulum. Now we will move on to second organelle. This is called the Golgi bodies. And they are having a lot of structural similarity. And if you see the Golgi bodies, the position of Golgi bodies, I'll just like to take you to the previous diagram once again. See, this is the nucleus. Outer to nucleus there is endoplasmic reticulum. Then on the next level, outer to endoplasmic reticulum, you can see Golgi bodies. Now we will come to the, just have a visualization in your mind that Golgi bodies are seen towards plasma membrane, and endoplasmic reticulum are something proximal to nuclear membrane. And this is the structure of Golgi apparatus. There also we can see different kinds of membrane bound structures. Whether it is Golgi bodies or endoplasmic reticulum, all are having unit membrane and all are lipoproteinaceous. You see the scanning electron microscope, you will see the endoplasmic reticulum like this. So these bodies are actually present in cells. So Golgi bodies is a complex, and it is made up of stack of membranous flattened sacs. And these flattened sacs are connected by interconnected tubules, and they are having vesicles and vacuoles. They are first discovered by Camillo Golgi, and it has synonyms like dictyosomes, Golgi bodies, lipochondrion, internal reticular apparatus, canalicular system, trophospongium. These are all different names of Golgi apparatus. The size of the Golgi complex varies between 1 to 3 mm to 0.5 micrometers depending on the age of the cell. When the cell is young, Golgi apparatus will be larger. When the cell is old, Golgi apparatus will be smaller. The shape of Golgi varies with different size, shape, or they can take different shapes depending on different cells. And they are seen towards nucleus. And often around the Golgi apparatus, there is a clear zone is observed within the cell, and this clear zone is called zone of exclusion. And the electron microscopic studies have revealed that Golgi apparatus has disc-shaped structures called cisternae, peripheral network of vacuoles, there are vesicles and tubules. Anyhow, all Golgi body membranes are trilaminar unit membranes and they are lipoproteinaceous. Coming to the first structure, cisternae. We have to be careful that in endoplasmic reticulum and Golgi bodies, cisternae is present. Cisternae are elongated, flattened sac-like structures. They are filled with some sort of fluid. They are stacked one above the other. You can see this, they are stacked one above the other. And this stack contains 2 to 8 cisternae unit, one above the other. And these are fixed in place or they are cemented with the help of a cementing substance called intercisternal material. So between these two cisternae, there is a material that is called intercisternal material. And in some species, some organisms, cisternae are having pores, and such cisternae are called fenestrated cisternae. And if you see this, the cisternae are slightly curved. One end of the cisternae is concave and the other end is convex. The concave side is pointing towards cytoplasm and the convex side is pointing towards nuclear membrane. Again, if you see the cisternae are having a curved structure. You can see there is a curve on the cisternae, and in this cisternae, one end is convex surface that is pointing towards nucleus, and the other end is pointing towards plasma membrane. And the convex side or the convex surface is called forming, new lamellae forming surface. They are the areas of new cisternae formation, and that is proximal to nuclear membrane. And because from the endoplasmic reticulum, often these lamellae are added to Golgi apparatus. The concave side is the maturing phase, and from that region, cisternae will degenerate into vacuoles and vesicles, and they will be released onto the cytoplasm. And that is cisternae. Second one is called vacuoles. Vacuoles are the large, spacious, round sacs present on the edge of the cisternae. As the cisternae become old, see this edge, these edges become larger and they will be pinched off. They are formed by the expansion of cisternae and that will be released. The cavity of cisternae is 60 to 200 angstrom. Coming to the third component is called vesicles. Vesicles are small droplet-like structures released off. See, this is the endoplasmic reticulum, and above that there is the nucleus, and this is the plasma membrane region. So from this, by the degradation or degeneration of the cisternae, small vesicles or small structures are released. They are called vesicles. And another one structure is there. You can see here the adjacent cisternae are connected together with the help of a branched structures, or there is a connection between adjacent cisternae with the help of a tube-like structures, and they are called tubules. And the tubules will help the transport of materials from cisternae, vacuoles and vesicles. So that is the interconnection or the interconnectivity between the different cellular structures are with the help of tubules. Coming to the functions of Golgi apparatus help the formation of acrosome in sperm head. They do the biogenesis of lysosome. They do cell wall formation. They do plasma membrane formation, help the plasma membrane formation, and they do involve in intercellular and intracellular transport, and of course the mucoid cell, the mucus secretion is helped by these organelles. So the similarities, both organelles have cisternae, tubules and vesicles, but in the case of Golgi apparatus they are having vacuoles, and functionally they are different, that you can see from the presentation. And thanks for your patient listening.